Genetics Lab at Baylor College of Medicine. I've known Dr. Smith for a long time. She was my mentor when I was a student, but she's still in her 30s.
Oh, thank you.
So without further ado, Dr. Smith.
Thank you, Peter. Thank you, Peter. It looks like Dr. Hu gave you a very good introduction to what we're going to be talking about today. So I think we're ahead of the game that you already know Robertsonian translocations and inversions and insertions. So what I try to do also is to give kind of a clinical bent to my lectures. So let's see. Oh, okay. And should we put it on
Full screen.
full screen? Yeah. All right, great, thank you. Okay, so I've just listed some of the objectives of these lectures today and next week here. So. I wanted to talk a little bit about the cell cycle and kind of go a little bit into, what the structure of the chromosome is in interphase versus in metaphase, which, like I said, I think you've got a pretty good background, but let's just get everybody on the same page. So there are really two major parts of the life cycle of a cell, cell division and then everything else. And when we talk in the cytogenetics lab about interphase cells, we're talking about cells that can be in either G1 or G2, or they could be going through DNA synthesis, but not in mitosis yet. So during interphase, the chromosomes really resemble fine fibers, and that's what you see on that bottom picture. It's an electron micrograph. They're extremely decondensed. That's when genes can be transcribed. That's when the metabolism of the cell can happen. But we can't recognize the chromosomes at that point, especially to study them, to look for those translocations and things like that. So the other thing to kind of keep in mind, especially when we're looking at some fish signals, is that interphase cells, the chromosome is either, the DNA's been synthesized, so it's one strand, or it hasn't been synthesized, so it's one strand of DNA, one molecule, or if it's gone through synthesis, it's two molecules of DNA held together at the centromere. And that can, like I said, when you're looking at fish signals in an interphase cell, that may come into play. But in preparation for cell division, and this is important for the cell so that the chromosomes can be transmitted from one cell to its daughter cells faithfully, the chromosomes become more and more condensed. And they'll eventually line up on the spindle apparatus. Oops. So prophase, before they've lined up, here's a metaphase plate. So the chromosomes actually under electron micrograph you can identify individual chromosomes, but they're all lined up together there. What we do in the cyto lab is to get them to spread out so we can count them and analyze them a little bit better. So basically what we're looking at is prometaphase or metaphase chromosomes in the cyto lab. So what this requires is to get a spread like this is a chemical that will disrupt that spindle apparatus. So that's part of the procedure of actually culturing and harvesting the cells. Yeah. So it sounds like you've gone through kind of what does a normal karyotype look like. But again, let's kind of just make sure everybody's on the same page. This is a normal male karyotype in a somatic cell, not a germ cell, with 46 chromosomes. And the chromosomes are numbered 1 through 22. Originally, it was based on size and centromere position, because when they first started looking at chromosomes in the 50s, they did not have a banding. We didn't know how to band at that point. So it was a very uniform stain all the way down the arm. So really the only thing they had to go on was the centromere position and the size of the chromosomes. The other thing I want to point out on this slide is that the X and the Y chromosome are obviously different chromosomes, but there are some what we call pseudo-autosomal regions on the top of the short arm and on the bottom of the long arm that have the same genes. So there are some genes on X and Y that there is a double dosage, so we have the same double dosage in the female. And just again, as I'm talking about some of the clinical things that we see in the Cyto lab, to get everybody on the same page, the centromere divides the chromosome into long and short arms. So short arm by convention is on the top, even if the centromere is pretty much in the middle of the chromosome, and which we call a metacentric chromosome. And so the short arm is designated P for petite. Q simply came after P in the alphabet, so that's why the long arm is known as the Q arm. Just kind of keep in mind that all chromosomes need telomeres, a cap to keep their ends from sticking to other things. We've got a submetacentric chromosome where the centromere is closer to one end than the other, and the acrocentric chromosomes that have stalks and satellites on them. So the acrocentric chromosomes, 13, 14, 15, 21, and 22, are the only chromosomes that should have those stalks and satellites, and those are areas of fragility, and that's why we see a higher incidence of Robertsonian translocations. The other thing other terminology would be homologous or homolog. So each of the pair are considered homologous to each other because they've got the same genes on them. The genes may be different alleles of each other. There could be some base pair differences, but it's still the same gene. And again, this is a schematic showing the metacentric, submetacentric, and acrocentric chromosomes. And these, usually when I draw diagrams, I usually use the chromosomes that have gone through DNA synthesis. So these are ready to divide. Yeah. Okay, so like I said, I mentioned the telomeres. The actual telomere itself is a 6 base pair cap. And this can be repeated 250 to 1,500 times, depending on the cell type. We also know that cancer cells have much longer telomeres. That helps them be immortal. Because as cells go through cell division, every passage in a normal non-cancerous cell, the telomeres shorten somewhat. So, but their purpose is to protect the structural integrity of the chromosomes. Without the telomeres, they could form rings, they could form dicentrics, so they have to have some sort of something to protect them from sticking to other things. Now, above that 6 base pair cap is a region that's called, it's telomere-associated repeat. So that's also a repetitive region, but it's not that actual cap. And then for each chromosome, the unique sequences begin at 100 to 300 kb in from the telomere. And this is where the unique DNA sequences are. That's a pretty gene-rich region. I guess when fish came into vogue, probably late 80s, early 90s, more clinical work was done with fish in the 90s. We had what we called a telomere panel, and those fish probes were unique to each chromosome arm, and they would probe to right there, right above that repeat. You may have also seen fish pictures of telomere fish. Those probes are going to be probing in this region of the repeats. Okay, so centromeres, besides dividing the chromosome between P and Q arms, it certainly has a function during cell division. So it's repetitive, it's non-coding, highly compact DNA. The predominant repetitive DNA in a human cell is alpha satellite DNA. Again, tandem repeats in this region, about 171 base pairs extending over a megabase or so. And centromere sizes do vary between chromosomes, and they vary from person to person for the same chromosome. Again, bringing centromere fish into it, we do see some size differences, and sometimes it can look like there may be 2 centromeres there, and when it's really only 1, it's just a really large one. So the stalks of the acrocentric chromosomes, actually, they are repetitive DNA, but they do code for something. So a lot of the repetitive DNA does not code for any genes, but the stalks actually code for 18S plus 28S ribosomal RNA. And that is the site that organizes the nucleolus during cell division. Okay. Excuse me. Okay, so to kind of talk a little bit about normal chromosome structure before we get into the clinical stuff. Each chromosome is composed of one linear molecule of DNA, and there are 2 different classes of proteins. The percentage of each component is roughly 33%. So collectively, when we're looking at a chromosome in metaphase, we are seeing both DNA and protein. Collectively, this is called chromatin. So when I write a clinical report to the doctor and I'm talking about a structural abnormality, I may talk about the chromatin from this chromosome. So when I say that, I'm talking about the whole structure. There are 5 major classes of histone proteins, and then there are non-histone proteins, and there are hundreds of thousands of different kinds. But what I'd like for you to appreciate is the variety of functions that those proteins serve. Some are simply structural. It's a very important role, so probably to say some serve only a structural role is kind of downplaying it a little bit too much. The structural role is very important. Others of these proteins function only during DNA replication, like DNA polymerase. Some function during chromosome segregation, like the kinetochore proteins around the centromere. And then the largest group of the non-histone proteins have a role in gene expression. And that's pretty much all I'm going to say about that. But I think I would like for you to appreciate the variety of functions there. Okay. So if you think about how much chromatin or how much DNA is in a cell, and we have to package all of that into the nucleus within the cell, it has to undergo multiple layers of folding and tucking and organization. So what they have discovered is that chromatin the DNA is wrapped around these histone proteins. So each little histone protein complex that is composed of H2A, 2B, 3, and 4 have about 160 base pairs of DNA wrapped around it a couple of times. Then there's linker DNA between this core and another core. And this is called a nucleosome. And then there's another protein that attaches to the DNA as it enters the nucleosome. Okay, then the nucleosomes, each chromosome contains several hundred to 1,000 to over 1 million nucleosomes, depending on the size of the chromosome. So it basically condenses the DNA at least 7-fold. But you can see these are it's kind of like beads on a chain, and they're not evenly spaced throughout the genome, but that placement evidently is very crucial for gene function. So it affects the access of the proteins to DNA, and thus it affects gene expression, transcription, all the functions that a gene has to do. The nucleosome placement is also transmitted with a high fidelity from through mitosis or meiosis, so from the parent cell to the daughter cell. So, and the other thing about this structure is it does change during the cell cycle. So it's what we call dynamic. Chromatin structure is dynamic, and that allows the protein access to the DNA during interphase. It permits the protein, it permits processes, such as gene transcription and replication to occur. So there is a kind of a method to the madness of all of the organization at the molecular level. And then just, we've got the nucleosomes. Those also have to be folded into some sort of structure. These are just a couple of models that I'm not sure that we know exactly for sure how, which model fits, or maybe there are other models that are actually more accurate. But we do know there has to be higher order of packaging to fit the DNA into the nucleus. And like I said, these are just several models that have been proposed. Okay. So that's kind of my foray into the molecular structure here. What I also want you to appreciate is that there are two major classes of chromatin. So euchromatin, heterochromatin. And the euchromatin does contain the protein coding genes. It also contains DNA that does not code for a particular protein, but it's still active in some way. And so an imbalance in the euchromatin often produces a phenotypic effect clinically. There are some euchromatic variants for which small imbalances seem to not have a phenotypic effect. A couple of that come to mind are on the short arm, the particular region on the short arm of chromosome 8. I think there's a region on the short arm of 5 also. But the euchromatin is opposed to heterochromatin, and there are two different types of heterochromatin. One is constitutive. So these are the repetitive DNA sequences which don't contain genes, and they're not transcribed. They remain condensed at all times in the vast majority of the time. Then there's facultative heterochromatin, and this is heterochromatin that can be euchromatin under a different circumstance. So it's been inactivated, and when it's inactive, it's called facultative heterochromatin. When it's active, it's called euchromatin. And an example of this is the inactive X in females, and we'll get to X inactivation. Okay, and then here's some examples of locations also of constitutive heterochromatin. Right under the centromere for chromosomes 1, 9, and 16, and then the distal long arm of the Y chromosome are all very good examples of heterochromatin, constitutive heterochromatin. And these vary in size. Sometimes we can see 9s that one 9 is extremely long because it's got a lot of heterochromatin in it, and its other homologue is really short. We now know when we see that in a clinical case that it's nothing to worry about, but if you imagine back when they first started looking at metaphase chromosomes, what is this? What does this mean? So fortunately, all that's been worked out. The short arms of the acrocentric chromosomes can certainly vary in size. Now, short arms of the other chromosomes, the metacentric and the submetacentric chromosomes, don't vary in size unless there's been an abnormality. But the short arms of the acrocentrics can vary in size, and it's considered polymorphism, or it's clinically insignificant variation. We can see double satellites. We can see, actually, I've seen on occasion an acrocentric without satellites. and then the length of the stalk, the number of ribosomal RNA genes in these stalks can vary. That doesn't have, again, a clinical impact because we've got multiple copies of it. The human body can handle multiple copies of those genes. It just doesn't transcribe what it doesn't need for the stalks. So let's talk about a little bit about X chromosome inactivation. So females have two X's, and as I mentioned, the pseudo-autosomal regions on XP and XP, YP, XQ, YQ, but in between there are a lot of genes on the X chromosome that that females have two copies of that males only have one copy of. And a lot of those genes, female doesn't need but one copy. So how the compensation is done by inactivating one X chromosome. So any individual that has two or more X's will have one or more of their X's inactivated. And that's the inactive X appears as a bar body, and this is an interphase cell with the from a female with a darkly staining body here that's called a bar body after the scientist who discovered it. It is, though, important for female development that both X's be active at conception, and we'll talk about Turner syndrome. Is there a question? Oh, no, that's okay. That's okay. Please feel free to stop me if you have any questions. We'll talk about Turner syndrome, where the females only have one X, and you'll see how that individual does not develop totally normally like a normal female. So the inactive X one X is inactivated fairly early in oogenesis. So the inactive X is turned back on during oogenesis, but so that both in a female fetus or a female embryo, both X's are active, but actually fairly early in embryogenesis, that X is inactivated. So, but like I said, very important in the beginning for it to be there. Females get it, one X from mom, one X from dad. Which one's inactivated? Well, for the most part, or in most cases, it's a random inactivation. And each cell that has two active X's that's going to inactivate one of those cells will basically choose which one to inactivate. And so it generally ends up that there's a fairly equal number of maternally derived X chromosomes that are active and paternally derived. Unless there's a problem, a mutation or something on one of the X's, and then we can see skewed inactivation, so that it's most of the X's from the mother are going to be inactivated because there's, say, a deleterious gene mutation on that X, as opposed to to the other one. But once a cell inactivates either the paternal or the maternal, all of its cells afterwards have the same X inactivated. So that follows that cell line all the way through to the death of the cells. And so in a female, you basically have all tissues that are a mixture of cells. There's a book called All Females Are Mosaic, because of the X inactivation. Now, how does this X inactivate itself? There's a gene in the XQ13 in this band right here called the EXIST gene, and that stands for X inactive specific transcript. And it produces RNA the RNA is not translated into a gene, but that RNA coats the X and helps silence it. That's not the only part of the mechanism. I mean, it's a fairly complicated process. There's some chromatin modifiers that change the structure, without that EXIST gene, inactivation will not occur. And it works on that particular chromosome, not on the other X. So even if the other X has an EXIST gene, it's not going to inactivate the one that doesn't. The EXIST gene itself is regulated by methylation, and this is a way in which a gene becomes inactive. Now, we also now know that at least 10 to 15% of genes on the X escape inactivation. There's one group, obviously, that's related to the genes on the Y chromosome, so the PAR1 and PAR2 regions, but there's another group that doesn't have related genes on Y. So there are some dosage differences, I think is fairly understandable between males and females. It's also, we found that the genes that do escape inactivation that are outside of PAR1 and PAR2 are clustered in domains, and a lot of those are on the short arm of the chromosome, so short arm of the X, sorry. And that comes into play, this comes into play with some of the variant karyotypes, again, that we'll see in Turner syndrome. Okay, so just a little bit about DNA replication. The double helix unwinds, and each strand serves as a template for the new molecule. The protein that helps add those nucleotides, those base pairs, is DNA polymerase. And it typically does that at a rate of about 50 per second in a human cell. Synthesis does occur from in one direction only, 5' to 3'. And so they call one the strand that goes 5' to 3', the leading strand, and it can just be synthesized straight down. But the other that actually runs in the opposite direction, there have to be blocks of replication that start and then a joining of those. So, so there actually are, and there are multiple origins of replication starting, approximately 10,000 in mammals. If there was one starting point for replication, it would take approximately 800 hours for DNA to replicate itself. So having multiple replication sites kind of speeds up the process, basically. And so each DNA segment has its own characteristic timing of replication. Heterochromatin is typically late replicating. That they used to use that late replication of the X chromosome as a way to identify and there are some other molecular methods now that we can use to identify the inactive X, but at one point it was having to culture the cells to see which one was inactive. So then just kind of going on, telomerase is another protein that's in the nucleus that's required to complete replication of the telomere. So it doesn't just start at one end and go to the other, include the telomeres. The telomeres have to be replicated as well. So the lower the telomerase activity, you have progressive shortening of the telomeres with each cell division. And like I said, with cancer cells and having longer telomeres, they also often have a high telomerase activity. But DNA replication is only one part of the process. The proteins have to be replicated as well. So that's also another a piece to that puzzle that has to be done faithfully. So I think you may have talked about mitosis and meiosis because you just seeing what your test questions were. So I won't spend a lot of time on this. Just kind of again, during mitosis, the chromosomes that have been replicated, the sister chromatids separate to opposite poles. Hopefully, obviously with non-disjunction, that doesn't always happen. And there, again, other proteins like cohesin that hold those sister chromatids together until it is degraded so they can separate during anaphase. Oh, okay. Okay. All right. So, kind of have all that as a background. In the cytogenetics lab, in the clinical cytogenetics world, or clinical genetics world, chromosome abnormalities are a major contributor to some morbidity patient morbidity. So there are various, obviously, types of chromosome abnormalities. We'll look at both numerical and structural abnormalities. There's also a class of abnormalities called uniparental disomy, that both homologs are from the same parent instead of having biparental inheritance of each homolog. That's something that we can't look at on a chromosome spread and say, oh yeah, that one, obviously we can say that's from mom and that's from dad, or both of those are from mom. It would be a molecular technique, but I like to talk about it as a chromosome abnormality. The other thing, we're going to obviously mostly be talking about constitutional abnormalities, not the somatic ones that are come up for cancer. So the methodologies, what are the typical methods that we use in the cytogenetics lab? Well, the conventional karyotype, you've seen a karyotype, that's the conventional cytogenetics, conventional chromosome analysis, G-banding analysis is called various and different things. We have to have live sample, so we can culture the cells, because only live cells can go through cell division, and that's where we have to look at, we need the cells from the metaphases to, or the chromosomes from the metaphases to actually do a karyotype. So with the conventional analysis, we're going to look at numerical abnormalities, we're going to look at some balanced structural abnormalities, and you've already been introduced to the fact that we can't see everything on a karyotype that can be seen molecularly. And so we look at both balanced and unbalanced structural abnormalities with karyotypes. Now, alluded to FISH a while ago, fluorescence in situ hybridization, more commonly known as FISH. And what it does is detect specific abnormalities. So the use of FISH would be dependent upon on the availability of the probe. There are companies that make the probes, so there are many commercially available probes. If you've got a laboratory that has access to bat clones, then you can also make your own probes. But this also requires time and effort to do it. But the use of FISH, and like I said, that came, I guess, karyotyping, the first karyotypes were actually produced in the late 50s, and that's when they discovered that Down syndrome was trisomy 21 and a couple of other syndromes. FISH came in, banding came in the early 70s, FISH then came in to use in the 90s, and then chromosomal microarray in the 2000s. What FISH did was allow for the detection of some abnormalities that were too small to be seen by cytogenetics. But you kind of had to know, okay, what to fish for. So this is just showing how you can, the probe, there's a piece of DNA that is labeled with a fluorescent probe, and then we can either put that on, apply that to interphase cells or to metaphase cells. And then chromosomal microarray uses extracted DNA. It can detect numerical abnormalities. It can detect unbalanced structural abnormalities. Our platform detects small, well, actually, we reported out deletions that were a KB or less because it was an exonic deletion. So you've got to have a platform that will allow that, but it's certainly that is the power of microarray analysis, to actually detect one exon deletion from a gene. It also can, depending on the type of probes used, can detect some cases of uniparental disomy, but not all of them. What microarray will not do, will not detect balanced structural rearrangements. And it can't determine the exact nature of the rearrangement. What we report out on microarray analysis is copy number gain, copy number loss. Okay? The knowledge of the structure of that is important for recurrence risk to give the parents. So this is just a little bit more about how a microarray is constructed. There are hundreds of thousands of pieces of DNA that are put on a slide, and some of them look like microscope slides. Some are little chips that go look like little cassettes that go into the laser scanner. But each area of the represents sequences throughout the genome. And each spot represents a genomic sequence, and that sequence that's maybe in this spot here is going to be repeated over here. So we've usually got we've got repetitive sequences on there just to increase the power of the testing. But basically, the DNA is extracted from the patient, labeled with a fluorescent dye, again, kind of like fish, and then hybridized to these probes on the slide. With array comparative genomic hybridization, we do the control DNA along with the patient DNA. On other platforms, the patient DNA is the only one that's labeled. It's run through a scanner, and then it's compared to a database that has the control. So an array is always compared to controlled DNA. And when the patient it has a higher intensity at that probe level, like in this middle here, that's telling us there's a gain of DNA, and then when there's less intensity as compared to the control, it plots below the log ratio line of 0 here, and that's telling us that there's a deletion in that area. And then the arrays can also give us the exact base pair, or fairly close to exact base pairs of what's deleted or what's gained, what the boundaries are. Okay, so this is just a comprehensive list of how mechanisms that chromosome abnormalities come from. So I'm not going to read through the whole list. We'll talk about a lot of these as we go through some of the syndromes. But I just wanted to get this all in one place. And so, and you can see there's quite a few of ways that an abnormal phenotype can come about. Okay. Okay, let's kind of get into this, and then maybe in about 15 minutes we'll take a break, if that's okay. So aneuploidy, gain or loss of a chromosome, that affects about 4 to 5% of all clinically recognized pregnancies. So if you take just a general newborn population, less than 1% will have a trisomy or monosomy. About 4% of stillbirths, though, can be affected by aneuploidy, but most aneuploidies end in miscarriage. And if you just look at overall miscarriage from first trimester to third trimester, greater than 35% of the miscarriages are shown to be aneuploid. The vast majority of those are going to be lost in the first and early second trimester. Trisomies are the most common group of abnormalities seen in these studies on tissue from miscarriages, 16, 22, 21, and 15. But all chromosomal trisomies, including for chromosome 1, the largest chromosome, have been detected in a miscarriage sample. So the trisomies that are viable, and when we say viable, it means compatible with live birth, there are only 3. Trisomy 13, 18, and 21. Trisomy 13 and 18, as you will see, are those babies do not live long if all cells in their body are trisomic, but they are compatible with live birth, unlike a trisomy 16 conception will not make it to term. So, and here's again, I think you've been introduced to the concept of mosaicism. So many of the autosomal trisomies are viable in a mosaic form. Some of the well-known ones, well described in the genetics literature, trisomy 8 and trisomy 9. I've had a lot of experience in prenatal diagnosis with seeing mosaic trisomy 15, mosaic trisomy 7. So trisomy 21 is the most common. This is the one that's associated with Down syndrome. And approximately 95% of patients with Down syndrome will have the trisomy 21 form. Okay. Approximately 1 to 2% have cells with 3 copies of 21 and cells with the normal, so they're mosaic. And then up to 4% will have the structural abnormality, the Robertsonian translocations that you've already been introduced to. So there are 2 different mechanisms that will produce mosaicism, mitotic error, non-disjunction, and then there's also trisomy rescue, which we'll see in a bit. So trisomy 21, probably many of you either have a family member with Down syndrome or know someone who does. Like I said, it is one of the most common, overall one of the most common chromosome disorders, with a frequency in the general population of around 1 in 700 to 1 in 800 live births. So some of the features are hypotonia as an infant, so low muscle tone, some delayed developmental milestones, and when for those of you who have children, you were when did your child first sit up? When did they start walking? When did they start talking? These are all delayed somewhat in Down syndrome, but it's certainly all milestones will be met. So the phenotype of not only Down syndrome, but the other syndromes that we'll talk about are features that are seen in the general population, isolated in the general population a lot, but they come together in the patient with Down syndrome or with trisomy 18 or whatever to form that phenotype. And so, like I said, each feature can occur as an isolated event in the general population. And you'll also see some of the features kind of common among chromosomal syndromes. So the upslanting eyes, the white specks and the blue iris of the blue-eyed babies, called Brushville spots, kind of a flattened nasal bridge as opposed to what the parental phenotype looks like. Instead of 2 lines, separated lines, some people call them lifelines, they often have a single palmar crease. This is actually seen in over 1% of the general population as well. This little baby showing a bit of a small mouth with protruding tongue. As she grows, that will resolve. The epicanthic folds around the eyes, it does not affect the vision. They're shorter than the chromosomally normal siblings. Again, all height has to be kind of looked at by what is the parental height or mean parental height, what's the family height versus what does the extra chromosome do. So, but they will usually be shorter than other family members. The major problem, none of those issues cause any clinical problems for them. The main thing, besides the delay, would be the cardiac malformations and GI malformations that could be very serious and need immediate attention when the baby's born. They do have an increased risk of leukemia. The leukemia that children with Down syndrome get more often than children without Down syndrome is a megakaryocytic leukemia, the myeloid cell line, as opposed to the lymphoid cell line. So children without the extra 21. When you hear childhood ALL, that's acute lymphocytic leukemia. That is by far the most common in the general pediatric population. But these kids tend to get one of the more adult-like leukemias. The names of them?
Yes.
Okay. Acute lymphocytic leukemia, that's of the B, mostly B cells. And you'll get all this in hematology as well. And then acute myeloid, which I said megakaryocytic, but just it's acute myeloid. It's the myeloid cell line. Okay. You're welcome. Okay. And then there's an overall life expectancy that's somewhat shortened. Compared to the chromosomally normal siblings, the severe heart defect, if their heart defects can be very severe or very minor. Obviously, the more severe heart defects would have more of an effect on, negative effect on survival. But survival currently well into middle age, I've even seen sixth decade of life, so into their 50s is actually becoming more and more common. Better health care, and one of the medical students I was talking to a couple weeks ago, we kind of figured also that 50 years ago, these children were probably more often than not put into institutions, and that I don't think was a very good health, or it definitely had a risk for health issues in that. They are more prone to infection, but again, better health care, being cared for at home is going to definitely help them. Okay. So we'll come back to Robertsonian translocations in a bit. So trisomy 18, it's also known as Edwards syndrome, but unlike Down syndrome, you usually don't hear the term Edwards syndrome, you hear trisomy 18. You can see that obviously we've got a much bigger chromosome, so there is a good bit more impairment, but it's not nearly as common in live birth. It's much more common at conception. So the estimates are with Down syndrome conceptions, maybe 25 to 30% of them will they will be lost prenatally through miscarriage. The estimates are 95% for trisomy 18. They are very severely growth retarded. There's prenatal onset of that. There's failure to thrive after birth. They're profoundly neurologically impaired. So physical and mental development severely impaired. They're also very hypertonic. The young babies with Down syndrome are hypotonic. These kids are hypertonic, so that you see joint contractures. And this can be seen in utero on ultrasound too. So we occasionally will get an amnio or prenatal study on a baby fetus that, and one of the features that they list from the ultrasound is arthrogryposis, and that's just simply a term for joint contractures. This clenched fist, the elbows, the knees, the wrist, ankles, that's where you see this hypertonicity. They also have another feature that they also can see on trisomy 13, but the rocker bottom feet. So it's kind of like the bottom of a rocking chair. And this has been seen on ultrasound as well. So these are just some other features that are associated with trisomy 18. The cardiac defects, abdominal wall defects, diaphragmatic hernia where the hernia is not completely formed, renal, GI, spina bifida, so interruption in the spine. It's a neural tube defect where the part of the spine did not develop or it's outside the body. So some pretty severe malformations. Their survival is extremely compromised unless the baby's mosaic. unless they have a normal cell line. So, and even with the normal cell line, there is going to be some neurological impairment. Okay, let's do trisomy 13, then we'll take a break. Okay, so again, you can see how much larger the chromosome, that 13 is than the 21. So this one is quite, the phenotype is one that, somebody would, a layman would look at the baby and go, no, and immediately know something's wrong. Again, the name of the physician who first described it, but usually it's called trisomy 13. Incidence in the live-born population, very, fortunately not very common. Like Down syndrome, though, this is an acrocentric chromosome. So the majority are trisomic form, but there's also Robertsonian translocations. And here, 80% due to trisomy 13, 20% due to the translocation, whereas with Down syndrome, it's 95%, or 95, 96% for trisomy, only 4% for the translocations. Major craniofacial anomalies, microcephaly, brain anomalies. The holoprosencephaly is instead of the brain being in having two lobes, they can be complete or incomplete holoprosencephaly where it's really starting to look just totally like one lobe. They have some very severe features otherwise, and again, profound neurological impairment. And we're talking survival, median survival, unless they're mosaic of, days instead of weeks or months. Okay. All right, so why don't we take about a 15-minute break, and then we'll come back and finish up. You want to stop the recording? Okay. I'm glad you know how to do this.